All right, today we are going to talk about the endoplasmic reticulum, and we are going to be covering a few points about this. The first one is let's talk about what the roles of the smooth and rough ER, what they are, and how they're different. So, the endoplasmic reticulum accounts for more than half of the membrane within the eukaryotic cells, and it is also the storehouse of that membrane material. And as we already know, the ER is continuous with the nuclear envelope. Now, the endoplasmic reticulum can be divided up into two very distinct regions. We have the smooth ER, which lacks ribosomes, and we have the rough ER, which appears under the microscope to be studded with ribosomes. So here is some electron microscopy images of these differences here. This is the smooth ER, and you can see that you see little bits of membrane, but it's very smooth in appearance, to where over in the rough ER, you can see these little black dots, which are actually ribosomes, which are bound to the surface. So this clear white area is the lumen of the ER, and you can see that there are ribosomes bound to the outside. And this region, this darker region in between, is continuous with the rest of the cytoplasm. All right, so the smooth ER has several jobs. One of its big ones is to synthesize the lipids. So all of that membrane material originates from the smooth ER. But it also serves some other functions as well, such as metabolizing carbohydrates, detoxifying drugs and poisons, and also it acts as a storehouse for calcium ions. So, as we'll see here in a moment when we talk about this lipid synthesis, you'll notice that the inside of the ER can serve as a holding place for particular enzymes. So by shoving all those enzymes into one spot, it can carry out a particular task very well. And as you'll see later on, there is a lot of carbohydrate manipulation that is happening within the ER. And so it makes sense to do a lot of that metabolism here and shunt a lot of those carbohydrates into this space. In terms of detoxifying drugs and poisons, this is also because some of the same enzymes that process lipids can process other things as well. And largely what they can do is make very hydrophobic toxins more hydrophilic. And once you have a toxin change from hydrophobic into hydrophilic, it then becomes possible to secrete that out in the urine. So this is something that takes place here utilizing a lot of the same enzymes that are used for lipid synthesis. And in terms of calcium storage, you'll find out later on when we talk about cell signaling, calcium can serve as a signal inside of the cytoplasm. So if we're using calcium as a signal in the cytoplasm, it means we need to store it somewhere else. And the ER becomes that spot where we can put it and keep it away from the signaling mechanisms until it's needed. So let's start off talking about synthesis of lipids. So typically what happens is there are membrane-associated enzymes. There are some integral membrane proteins, some that are peripheral membrane proteins, but they tend to be associated with the membrane itself. And they will take glycerol 3-phosphate, which you should at this point recognize as one of the things that is produced during the breakdown of glucose. So this is one of the reactants that we see fall out of that, fall out of the glycolysis. We also have these fatty acid chains, which are linked onto coenzyme A. This is the same coenzyme A that we see used in the TCA cycle. So we have two fatty acid chains on these carrier molecules, and we have glycerol 3-phosphate. All of these things come together in the enzyme, which isn't shown, to produce this thing called phosphatidic acid. And this is essentially two fatty acid chains attached to what is essentially glycerol, but it's phosphorylated, the phosphate group being a remnant of where it was coming out in glycolysis. First thing that happens is that phosphate is hydrolyzed off, and so now we have diacetylglycerol. And this is the foundation for all of our various phospholipids in our membranes. At this point, this can be acted on by one of several different enzymes. One shown here is taking the molecule choline, which is attached to a CDP carrier. So we've seen ATP carriers before, we've seen GTP used in circumstances. Now here's an example of CDP being used as a carrier molecule, doing something different than just being a nucleotide. So here it is acting as a carrier model, and then we can attach that choline onto the group, and we see the addition of a phosphate in that process. The phosphate is coming from the nucleotide itself. This is, of course, not the only outcome. This is getting phosphatidylcholine, but we have all these other phospholipids which we can produce. And each one of these would have a different enzyme that is taking care of the role of this last step here. Each one coming in, putting in a different head group onto it. This is all happening on the cytosolic phase of the smooth ER. So you can see here we have the original membrane, and then as phospholipid synthesis occurs, we get an accumulation of newly synthesized lipids on this leaflet of the bilayer. And then some of these are specifically moved to the other surface using an enzyme called flippase. Okay, so flippase is responsible for taking one of these and flipping it to the other surface. Okay, recognize that this is something that would require some energy because we are essentially taking a hydrophilic head group and trying to bury it inside of a membrane, okay, or in this very hydrophobic space. Something that the water in the cytoplasm doesn't want to let go of that hydrophilic head group. So this is something that requires some energy. Now, not only do these flipases accomplish this, but they also are very selective. And this can lead to having different components or different phospholipids in different parts of the leaflets, right? So here what we're looking at is we're actually looking at the cellular membrane, and we're looking at the outer leaflet versus the inner leaflet here in yellow. So you can see most of the phosphatidylcholine is in the outer leaflet, to where most of the phosphatidylethylamine is on the interior. And you can see those differences. This is accomplished because the flipases are actively selecting which phospholipid they are going to flip to the other surface. So they are moving specific ones to the other surface in order to make it the way it appears. And this actually, as you'll find out, will play into how we how some of the chemical charges come about within the cell. So the inside of a cell tends to have a negative charge just underneath the surface of the membrane, and this is achieved through this act of flipping. All right, so moving on to the functions of the rough ER, it does have ribosomes bound, and it is secreting those proteins to the interior of the rough ER. It is in the interior of the rough ER that carbohydrates are added on to proteins, making lipoproteins, okay, which is a protein with covalently bonded carbohydrates. And we'll talk about the purpose of these later on. We'll talk in detail about that. But this is where that process takes place. The ER is also responsible for producing transport vesicles, which will allow proteins to move from one compartment to another, moving to the Golgi apparatus or moving to other areas of the cell. So now we kind of roughly know what the roles of the rough and smooth ER are. Let's take a moment to look at the secretory pathway. So here is a picture of a cell, and this cell has a very polar appearance. So one surface of the cell looks very different from another surface of the cell. And this is something that you would see when you have something like an epithelium, right? So if you have a layer of cells that are separating, say, the inside of the gut or the inside of the lung from the inside of the body, it would have this polarized appearance. So there was an experiment that was done where we radio-labeled proteins that were being produced, and then we took samples at different time courses, separated out some of these components to try and figure out where the newly produced proteins were. And what we found is that very early on, all of those radio-labeled proteins are appearing in the rough ER. After about 7 minutes, we find that those have moved on to the Golgi apparatus. So here we have hard evidence showing that they start off here and move to here. And then further on, if we wait even longer, we'll see these accumulating in the secretory vesicles, and then ultimately we see them secreted into the exterior. So the general flow is coming from the cytoplasm, or we have messenger RNA that is being produced in the nucleus that comes out of the nuclear pores. They come into contact with ribosomes. Those ribosomes associate with the rough ER and start making the protein, secreting them into the interior. Those then get transported to the Golgi apparatus, which ultimately get exported into the secretory vesicles. This is, of course, an oversimplification. There are pathways that allow us to bring things in from the surface, directing them into the Golgi apparatus, and maybe even then shuttling them to the other side of the cell. We have ways of going from one surface all the way to the other surface without necessarily going through the trans Golgi network at all. So, recognize that this simplified pathway is an oversimplification, but just remember that things are starting off in the ER, moving to the trans Golgi apparatus, secretory vesicles, and then exterior. And this will help you keep straight how the flow of material is going.